Whether it is plants or animals, they are all composed of cells, and the cell is the basic unit of life. A cell as a unit can digest material, can take food, can degrade the material, produce energy, can excrete the wastes. A cell can grow, a cell can move, it can occupy a position. And all the information required for the activity or the life process of the cell are encoded in the genes, and the genes are made up of DNA. When a cell is to double or divide to give rise to two cells, it must therefore also duplicate its genes so that each daughter cell gets equal amount of DNA or genetic information. In the cells, there exist catalysts or enzymes called DNA polymerases. A DNA polymerase is an enzyme which can look at the pre-existing DNA in the cell and make a copy of it so that the daughter cell will get the same genetic information. Unlike the animal or plant cells, there are also small organisms like viruses, and the viruses can also utilize the same mechanism for duplicating their genetic information. Some viruses have the genetic information in form of DNA, some have in RNA. Recently we know that there has been great deal of interest in a very particular kind of virus called as AIDS virus or human immunodeficiency virus. This virus infects the cell. It has, however, as genome RNA. Nevertheless, the enzyme which allows copying this molecule is also a DNA polymerase which can utilize the RNA. We have here with us today Professor Mukund Modak from University Medical Center of New Jersey. He is their professor in biochemistry, and Professor Modak has spent great deal of time, last almost 20 years, working on DNA polymerases, isolating the enzyme, looking at its structure, its function, how it's active in a cell. It's really a pleasure to have you, Professor Modak, here. And can you tell us, how does this AIDS virus really activate a cell? How does it infect the cell? And how can it therefore duplicate its genome, particular genome? And how can it propagate itself?
Well, as you know, every living thing has two functions. One, to live, and second, to divide or eat, or somebody has called meet and mate. That was the old theory. So the viruses are no exception to that. They go through the same thing. They eat food, and next thing you know, they are dividing. So AIDS virus, for that matter, has developed itself in a very nice, very cute way. In a sense, normally if there is an infection of a body, infection of a cell, body will fight that infection. But this virus infects the very cells which are going to fight that infection.
In other words, this AIDS virus is not really a true unit of life, but it's a parasite.
Exactly.
So can you tell us a bit about that?
Yes. This virus will depend lot on cell which it infects. It will take lot of food, lot of enzymes, lot of other proteins from it. Yet when it comes to duplication of its genetic material, it would not trust host. It will carry its own enzyme, which is a DNA polymerase or reverse transcriptase. But to really understand, let's quickly look how this virus infection occurs, or what's the life cycle of this virus. Okay. So if for a minute, if you pay attention to this figure here that I have drawn, you will see that virus has outside those bulb like things, which is what it uses to recognize which cell to infect. For example, you know that HIV virus infects immune cells, or specifically T lymphocytes. Now T lymphocytes on their surface have these blobs or have these holes in which those HIV virus balls nicely fit. Now using that mechanism, in a way, the virus find its target or find its host. So, once it goes in, first thing it does is as soon as it touches the cell, as soon as it finds its pocket to lodge that ball, it will uncoat, throw away that coat now and enter the cell. What is entering the cell is only two parts of the virus. You see, virus has its genetic material, which in this case is RNA, and it carries its own enzyme to duplicate that. Now, that is the crucial part here. It is carrying its own enzyme. What will that enzyme do? That enzyme will take that genetic material, which is RNA, and convert it into DNA, with the final purpose, ultimate goal is take that DNA and put it along with host cell DNA. Once it achieves that, it becomes one with the host.
Therefore, the objective is to convert the genetic information, which is in form of DNA, RNA, into DNA first.
Exactly.
And then only that DNA can be completely inserted in the chromosome of the host cell.
Exactly. Thereby, virus achieves immortality for that matter. Once it is inside the nucleus, once it is inside along with the host cell DNA, it is immortal. Now, to propagate further, it follows your central dogma of information transfer, namely from DNA it makes RNA, then it makes proteins. Now, as you recall, the genetic material is RNA itself. So, as soon as it makes RNA back from that DNA and that some part of RNA is converted to proteins, a new virus can form. And if you want to really see an animated view of this, I have a number of interesting pictures here.
Oh, that will be nice. But tell me, is this therefore this virus must be very small as compared to the cell itself?
Oh, I would love to show you that. How
Oh.
when people talk of viruses, people talk of cells. Like we look at our cells and we think, how many cells do we have? Billions and billions of cells. So, what's one lymphocyte in it?
That's
Extremely small. And how big is a virus? That's even million-fold smaller than that. But if you see now, just look at this picture, you will get a clear idea. Here is the electron micrographic picture of a T cell, the big wavy surface that you are looking at. That is how these T cells look. They are hairy cells, there are lots of hairs on them. You can't see them, of course, with naked eye, you need electron microscope to see that. But look at those tiny balls, those are the viruses.
I see.
See, those viruses are going to find their sockets within those structures. Look at this now, this is more amplified picture. Just the virus is shown here and the cell surface is shown. The virus has inside, if you carefully look at it, a strand of RNA,
Yes.
which is its genetic material. So, upon touching up that virus to cell surface, see the virus does not go, entire virus does not go in, it only sends in its RNA and enzyme.
Right.
Now, once it enters inside the cell or inside the cell cytoplasm, what you are looking at is again just two strands of RNA, and on that is two tiny enzyme molecules.
So, the enzyme is already in the virus to begin with.
Absolutely.
Yes.
The virus absolutely makes sure that it does not depend on host to make copy of its genetic
It
material.
uses its own enzyme.
It's to ensure that there is no damage to that.
Right.
So, that's one basic thing. Now, next process if you see, what you will see is that RNA is now extended to become just a single stranded RNA.
Yes.
Mind you, it is a single stranded RNA, whereas our genetic material DNA is double stranded.
Yes.
So, it is the task of the enzyme to take single stranded RNA
Yes.
and convert it into double stranded DNA.
Yes.
And how it is going to do is it takes that single stranded RNA as you see in this picture, then it uses substrates, food material needed from the cell itself.
You mean nucleotides?
Nucleotides, precisely.
Yes.
Nucleotide substrates that are used to build that DNA. DNA is composed of 4 nucleotides, as you know, ATGC.
Yes.
So, those are the very nucleotides it's going to take from the cell itself
Yes.
and weave them into a chain, long chain called DNA. So, it makes now from single stranded RNA to what you will call double stranded molecule, one of which is RNA, one of which is DNA.
That's right.
Okay, so here you are looking at one strand of RNA, one strand of DNA.
Yes.
Okay. Now, you have to convert this into 2 stranded DNA.
Yes.
In effect, you have to remove that RNA
Yes.
and then convert that RNA part into DNA. Okay. So, this enzyme has additional activity. See, enzymes are machines, they can do things very fast. So, it will have ability to remove that RNA, but RNA it will remove only when it has made that first strand of DNA.
Yes.
So, it is actually called DNA RNA hybrid.
Yes.
So, it is only when there is a hybrid that this enzyme will remove RNA part and actually synthesize using again those nucleotides second strand of DNA. And if you see that DNA now shown here as a red molecule,
Yes.
just to distinguish from the green molecules of DNA,
Yes.
it is not that DNA of the host is green color and virus of red color.
You have painted it.
Yes, absolutely.
For the sake of
clarity.
clarity.
And also red color, you know, is a danger. Well, this is the virus which has come inside your cell, so it is a dangerous thing to have,
Yes.
but you can't do anything. Once this has happened, this is what we call immortalized state. Now, the enzyme which did it was reverse transcriptase, okay, and that is the enzyme we want to worry over. Why?
So you have the RNA molecule introduced, it is copied into a DNA, one strand. So you have a RNA DNA hybrid. Now the RNA part of that hybrid is destroyed and the remaining newly synthesized DNA strand is made double stranded.
Absolutely.
And once you have this structure, only then it can go and insert itself in the host double stranded DNA.
Exactly.
And then become part of it.
Yes.
And therefore immortalized.
Immortalized. Now,
Very
you will say
interesting.
how big a part of DNA is it making? Well, if you consider this as a hundreds of million bits and pieces of DNA,
Yes.
this may constitute maybe 2 pieces, 3 pieces out of that.
So therefore, if a human has about say 50,000 genes, then this will be just one of them, very small part of the total.
Exactly, just one gene, you may even call it just 1 out of 1 million genes.
But one extra gene.
Yes,
Yes.
nonetheless. But human cell will not recognize that it is carrying this extra burden.
It will never know.
Never know, and so it will continue using, consider it as if it's its own gene, and therefore it will produce its products.
But actually this molecule which is inserted, is it really one gene or 3 fewer but still a small number of genes, more than one?
Actually, it's there are 5 or 6 little tiny genes which are required for virus. One of them makes code for virus, one of them makes this enzyme reverse transcriptase,
Yes.
one of them makes something to protect that reverse transcriptase and RNA,
Yes.
and that's about it.
Right.
It doesn't need any, everything else that's needed, it takes without asking host, shall I take it, shall I not take it? No, it doesn't need its permission. It's in there and it has developed a mechanism that it will preferentially take it. And as you know, Atithi Devo Bhava, whatever you say, the virus is guest there and host offers little bit room for it and it takes over the entire cell, begins to multiply, in the process destroys the cell, and number of new virus particles are released. Now, these virus particles are seeking another host in the same body, same human body.
Yes.
The net result is that you have depletion of your lymphocytes. Lymphocytes are your fighting cells.
Yes.
If you deplete them, you won't have anything to fight with. So, bacterial infection, other virus infections can occur rather easily.
Yeah.
Fungus infection can occur, and that's where the patient succumbs really.
So you see, therefore the one of the fundamental issues is, since we are all 3-dimensional structures, are these small proteins of viruses as well as these enzymes, are they also 3-dimensional structures? How they really operate at the molecular level to find the particular RNA molecule to make a copy? Can you tell us something about this structure?
Yes, this is actually interesting. You know, as we go down to the smallest level, we begin to think, is the world different? And it comes down, it's not different, it's almost same. We as a human body has an organization. We have a nose for breathing, we have hands for grabbing things, we have mouth for eating. Enzymes are almost made up of the same thing. It has a 3-dimensional structure. What we define right now, our ability to define them is somewhat not reached to a stage where we can quickly take enzyme protein and say, oh, this is how it looks. What we define them as a chain of amino acids,
Yes.
simply put. Now, chain of amino acid is simply one amino acid attached to other to other to other. For example, let me take this example here. Here is your amino acid number 1, 2, 3, 4, 5, 6, whatever 100 amino acids you have. Now, what makes individual enzyme or protein different? It's their folding. You see, these proteins can fold, make different kind of shapes and sizes. Now, how do they fold? It's the amino acid sequence as well as some genetic input from the DNA again,
Right.
indirectly put. We don't know exactly how this happens at this time, but we know that they finally take a specific shape or size or conformation. This is
Right.
what we will call conformation.
Right.
Meaning, does it have three-dimensional look? Does it have width, length, height, etc.? Yes, they do. And it's absolutely essential that that structure be assembled in that manner for a specific function to occur.
Yes.
Now, take example of a reverse transcriptase. Okay. It has such a kind of structure. How does one go about looking for these structures? Okay. First one has to do is purify this enzyme,
Yes.
meaning this is a protein.
Yes.
Take it from virus or take it from cells that have infected virus, or we can now cleverly use this new recombinant DNA technology,
Yes.
whereby genes coding for this enzyme protein can be selectively amplified, put into a bacterial virus, infect that virus, use that virus to infect bacteria and produce on large scale. As you know, bacteria grow.
So you can make a lot of protein
Yes,
of the same type.
precisely. Once
Then-
we have this protein now,
Yes.
then we can determine what is its amino acid sequence.
Yes.
Primary. Now that is only going to give you this info.
Yes.
Okay. We still have to know how it's folded.
Yes.
Now for that a technique called X-ray crystallography
Yes.
and molecular modeling
Yes.
is used.
Yes.
Okay. What essentially one does is allow this protein to make a crystal in solution.
Yes.
You put it in solution, take away water slowly,
Saturated solution.
Saturated solution.
You will crystallize the protein.
crystallize. Now if it crystallizes, what does crystallization means? It means it achieved a stable conformation.
Yes.
And is sitting. Now send X-ray rays through that at very small distances, a number of them, millions of X-ray diffractions you take. You get a shadow and generate something that you call a structure, a three-dimensional structure.
Yes.
Take for example this molecule. Look at this molecule here. This is something like a DNA type of molecule. It has width, it has height, it has depth to it. Now this can be seen when you send X-ray images through that, or X-rays are passed. Now what it gives you essentially is some outline,
A shadow diagram.
shadowed diagram. It's like take our chest X-ray and you see our bones,
That's right.
but you don't see any other thing. A task of ours now is take such a kind of structure and put meat on it, put these different organs on it.
Yes.
What does that mean? That simply means take your amino acid. Amino acid has a peptide bond, and amino acid has a side groups, chemical groups, CH2, 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 NH2, something like some are aminos, some are carboxyls. Those types of amino acid exist, and actually those are the ones which carry out enzyme function, because those can carry on a chemical reaction.
Yes.
After all, enzymes do carry on a chemical reaction,
Yes.
except that they do it at such a fast rate because of this conformation, because of the size and shape that they have achieved.
Yes.
And our purpose is to know by what kind of shape and structure do they recognize DNA. For example, if they want to duplicate DNA, they have to first go touch DNA,
Yeah.
then they will look at how to take nucleotides, how to fit the nucleotides,
Right.
get the exactly same DNA sequence.
Yes.
That's the key to it. For example, here you are seeing the three-dimensional structure of enzyme.
Yes.
Now, if you look on the left-hand side, or the red and yellow picture that you're looking at, those are the ones for HIV. Those are the ones for HIV reverse transcriptase.
Yes.
The other one is for bacterial enzyme, DNA polymerase. As you will see, both of these structures have kind of a hole in it. Now you will wonder, what does this hole do? Well, the DNA of this kind, if you consider DNA as a double-stranded molecule, can nicely fit into that hole.
So it's a sort of a tunnel for DNA molecule.
Exactly, you got that absolutely right. It goes inside. Inside it touches different ATGC bases and says, okay, what kind of copy am I making? Am I copying A? It's the base complementarity, meaning if it sees A, it puts in T nucleotide.
Yes.
If it sees G, it puts in C nucleotide. And it will keep doing that. As you see, this what you are seeing here is taken an X-ray structure,
Yes.
meaning simple X-ray figure
Yes.
on which different amino acids are mounted, completed their we call it side chains, or R groups,
Yes.
as the students might
The
know.
active groups.
Active groups. And that's how the structure works. Now we are going to recognize in this structure where does this nucleotide react, where does DNA react, where does extending chain reaction occurs. Here, take a specific example of HIV reverse transcriptase.
Yes.
Okay, HIV DNA polymerase from the viral enzyme. What does it have? Well, it's composed of 3 subunits. See, the blue one that you are looking at is where all this recognition occurs. The yellow one you are looking at is a supporting subunit, and the red part you are looking at, you remember back now we said that it makes hybrid and it must remove RNA.
Yes.
It's precisely for removal of RNA that part is used.
Right.
So it's very interesting that virus has all the things required in it, and it's the same enzyme now where you have heard of AZT,
Yes.
for example, a drug.
This is one of the drug used for treating AIDS.
Absolutely. This is perhaps the only drug right now which you can say is effective to some extent.
To some extent.
Okay. Why am I saying to some extent? If you look now what's happening to AZT, there are a number of patients becoming resistant to this drug. When we say resistant to this drug, that means this virus, instead of being stopped growing in presence of this drug, is now able to grow. Okay. So naturally, where does AZT go? AZT is one of the nucleotide. So its target is actually this enzyme, that it has to go and bind to this enzyme.
And then block the passage of everything else.
Exactly. So therefore, other nucleotides cannot go. Even if it goes into DNA, it blocks DNA right there, so that the DNA cannot grow.
Right.
Okay. And virus of course senses this somehow. See, as you said, every living thing, if you consider virus as a living thing, strives to survive. It's a struggle for survival. So it's going to come across and say, I must mutate. I must become something, change my structure.
You don't mean viruses have mind, do you?
I would imagine if you go deep down and if you are able to define mind, maybe they do. Whether they do or they don't, we see the result. That there are these enzymes which now are not affected by AZT.
So in other words, the virus is modifying its own enzyme structure such that it can no more be inhibited or affected by a drug like AZT.
Exactly.
So this is mutation is taking place.
Absolutely. And what we are trying to do now, one of the things that modern scientists are trying to do, is find where does AZT bind and how does mutation, now take a mutant enzyme, there will be some single change of one amino acid. Now where is that change and how does this effect takes place, the effect of mutation, meaning no more AZT sensitivity.
Yes.
Now by knowing where AZT binds and where normal nucleotide binds on this enzyme and how does this enzyme work, one can always build new inhibitors, because now this is in 3D. Remember, you know its depth, height, width, except your scale is atomic scale. You are not talking of 5 feet long and 3 feet high and 2 feet wide.
Therefore, by knowing the 3-dimensional structure of effector molecules such as this catalyst, which is so important for the perpetuation of AIDS genome, you can do engineering, you can engineer new drugs.
Absolutely. Yes.
Which can then, right from the structural point of view, fit at the right place.
Yes.
And either block the enzyme or unblock it possibly.
Exactly.
I see.
And we can even make new molecules, and because of this computer age, what we can do is we don't actually have to make the molecule first. We can design a molecule and say, is it fitting here? If it does, then you say it has to work as an inhibitor. If it doesn't fit, you can say chip this side off, take this off, take that group off. This is called organic synthesis. It will become such a powerful tool to guide what kind of inhibitors to make.
So tell me, about having looked at this 3-dimensional structure of this enzyme, which are the major features, characteristics of this, this body called enzyme?
What you will have to look at is, take just a gross look at it.
Yes.
You know that they have to bind DNA.
Yes.
So there is a big DNA binding track.
Okay.
You know, after all, this is not a small molecule.
Yes.
What you're looking at, this is a big molecule which is to be copied, and you are copying brick by brick, just this small unit at a time.
Yes.
Okay. So that's what you're looking at. So
Right.
therefore, this structure has a huge DNA binding track.
That's right.
Okay. You can also look at this structure in physical manner. Physical manner meaning ask the question, does it have electropositive charge, electronegative charge, how it is distributed on the surface. Now, if you take the look at this picture we have shown here, the red indicates negative charge and blue indicates positive charge. Now, you know that DNA is acid, deoxyribonucleic acid. Acid means it is negatively charged, and with it is going to bind to positively charged tracks. So, you are looking, yes, there exist absolutely good tracks here. Our task will be now to find out, going inside the structure, which amino acids made that track.
That's right.
Or is that just a track? Absolutely no amino acid by itself sits there. Similarly, there will be units, there will be place for binding these single units which are nucleotides.
Yes.
And those are going to be woven into thing, and those are the kinds of things we are doing with this structure of enzymes.
Yes.
Level, at that level, which we call atomic level structures now.
Yes.
And I am sure with this there will be a very basic understanding of how enzymes so faithfully makes a copy of the genetic material. See, this genetic material is billions of bases long.
So, basically if one is on stage and there is a dance festival, you can see the human dance.
Yes.
Or animal dance to that matter. And inside the cell you have molecular dance that is taking place. And for particular molecular dance to take place, hand in hand or different type of interactions, these molecules can change the shape, or they have defined shapes to allow certain types of hand in hand type of actions, interactions, and that is really what you are talking about basically.
Almost similarly, I mean, within human you have another human. If you want to humanize this enzyme, okay, by structure that you have seen so far, what you know is there is a basic body. For example, you have a hand. You can only extend your hand forward,
yes,
or on the sidewards, but you can't put your hand in the back. So, there are certain limitations on enzyme too. This structure tells us where those limitations are.
So, therefore, you have the molecule, say if this was a DNA polymerase which is dependent on DNA instead of reverse transcriptase, this enzyme will sit on this molecule and maybe crawl along the molecule making a copy.
Absolutely.
Or that the molecule will crawl along the enzyme.
Right.
It really does not matter,
yes,
which way it goes, but therefore, the enzyme structure must allow movement of one molecule relative to other one.
Precisely.
So that you can actually complete copying of
Yes.
one molecule into the other.
Inside the cell particularly, if you look, the DNA is more stable because it is bound to membranes, sites, etc. So, it is really the proteins which have to do the moving job.
Right.
But that does not mean they can't let DNA slide little bit here, there. So, there is nothing like fixed thing, it is a dynamic equilibrium.
This particular whole question about how enzymes operate, or DNA polymerases in particular operate, becomes more and more exciting. And the point is, larger the body you look at, easier it is to find out what parts do. Smaller the body that you look at, more difficult it is to discern parts of it, but when you do, it becomes logically very easy and simple to find out how these small bodies also can interact with other small bodies and produce something as complex as the copy of the total genome or the DNA molecule, which is an essential function for a cell before it can give rise to two, or for a virus to give rise to genomes for two viral particles, and therefore the multiplication process is facilitated. Naturally, the enzymes are catalysts, and the catalysts as they are, they still have to undergo same number of critical steps to carry out this particular final product of synthesizing DNA, or whether it's DNA or RNA, or at least duplication of the genome. It has been very nice to talk to you, Professor Modak, on this subject. This is something which is very exciting and really fathomless deep. It is going to the depth of the life processes. And I'm sure that there is a lot else to be done, and next time probably you'll tell us what is new coming up.
Oh, absolutely. We are all excited about it, and thank you for your time.
Thank you.